So I am Dr. Alice, Associate Professor, and I have 13 years of experience in the field of microbiology. So today I'm going to discuss about bacterial genetics via this Raaonline. So bacterial genetics talks about the genetic variations and the heredity that is seen in the bacteria. So when you take a bacterial genome, it has got chromosomal DNA as well as extra chromosomal genetic material. So that is the plasmid. And in this topic, we'll be talking about the genotypic and phenotypic variations, the various mutations, and the various gene transfer mechanisms, and also the significance of bacterial genetics in drug resistance, genetic engineering, and molecular genetics. So coming to the bacterial cell, it has got two components, that is extracellular as well as intracellular components. So the extracellular components includes the flagella, pili, fimbriae, capsule, slime layer, whereas the intracellular component consists of the bacterial chromosome. So since bacteria is a prokaryotic cell, it doesn't have a proper nuclear membrane, and so the bacterial chromosome is irregularly placed inside the cytoplasm, and it is called as the nucleoid. And the size of this bacterial DNA or the bacterial chromosome is 1 millimeter in length. So we all know the size of the bacterial cell is micrometer size. So the bacterial chromosome is supercoiled in such a way that it gets accommodated inside the micrometer sized bacterial cell. And it is haploid in nature, and some bacterial cells will have an extra chromosomal genetic material, that is plasmid. And some bacterial cell have 1 or 2 bacterial chromosomes. Example is Vibrio cholerae. So now coming to the structure of the DNA. So DNA is a double helix and it has a backbone which is made up of deoxyribose sugar and phosphate groups, which is interlinked by the nitrogenous bases where we have purines and pyrimidines. So we have adenine and guanine as purines and thymine and cytosine, the pyrimidines. And now, what is a nucleoside and what is a nucleotide? So nucleoside is made up of the ribose sugar and the nitrogenous base, whereas nucleotide is made up of the ribose sugar, the phosphate group, and the nitrogenous base. And now there is a pairing system that always exists. So adenine always pairs with thymine and cytosine always pairs with guanine. So now the thymine and adenine pairs with a double hydrogen bonding, whereas cytosine and guanine has triple hydrogen bonding. And also there exists this AT and GC ratio, and this is common between same bacterial species, whereas it varies with different bacterial species. And this determines the phenotypic and genotypic characteristics of the bacteria. So now coming to the structure of RNA, RNA will be single-stranded, and instead of thymine, we have uracil in place, and there are 3 types of RNA: messenger RNA, tRNA, and the rRNA. So now we all know about the central dogma of molecular biology, where DNA will be transcribed to RNA and RNA will be translated to proteins. So now when you take this translation process, it has 4 steps. So first is initiation, the second one is the elongation, and then the translocation, and then the termination. So now how do a prokaryote replicate? So it replicates by binary fission. And how does the nucleic acid replicate? So it follows 2 mechanisms. One is the bidirectional replication. So here the replication gets initiated at the replication fork. So replication fork is a place where unwinding of DNA happens. And now there are different enzymes that are responsible for the replication process. One is the helicase enzyme, which helps in the unwinding of the DNA. The second one is this topoisomerase that will prevent coiling or winding. And then the third one is the polymerase, which helps in the synthesis of DNA strand. And then the ligase, which helps to link the ligated fragments. And now this synthesis happens in the 5' to 3' direction, and the reading frame will happen in 3' to 5' direction. So this is called as bidirectional replication. The second one is the rolling circle replication. This happens in the case of conjugation. So now one strand will act as the template to synthesize the other strand. So this you call it as rolling circle replication type. So now what is a gene? So gene is a unit of heredity, and it is a segment of DNA that carries the nucleotide sequence and is responsible for the phenotypic and genotypic properties. So now these genes are made up of codons, and these codons are made up of 3 nucleotide bases. Okay, they are called as triplets, and they are present on the mRNA.